Our next speaker is Aykut Uren from Georgetown University. He'll be talking about CD99 inhibition in Ewing sarcoma. Did I get your pronunciation right?
That was perfect, thank you. And we had another Ewing sarcoma survivor. His name was Kevin Chen. He was a Georgetown undergrad. He had amputation just like yours, and he worked in the lab when he was undergrad. Then he worked in the lab as a research assistant for a year. Then he went to medical school at Georgetown. Now he's about to finish his residency at University of Southern California. So we have his spot open for you. So you're welcome to come to lab anytime. So this will be sort of an update for our recent CCF grant and what we've been doing with Ewing sarcoma and CD99 inhibitors that we're trying to find out. In general, when Big Pharma develops drugs, they usually do either through a high throughput screen or a structure-based design. So if you have the crystal structure of the drug, you can screen virtually in a computer environment. Or if there's an enzymatic assay, you can screen thousands, hundreds of thousands of compounds in these assays. But the rest is left for what they call undruggable, are actually the targets that we have a lot in cancers. And the targets that we like in Ewing sarcoma, they usually fall into this group. And Jeff will tell you about how we developed a drug for EWS fly. Today, I'll tell you one for CD99. Our unconventional approach to this new drug development has been we go after a target protein. So we select which protein we like to hit in a given disease. And then we identify small molecules that directly bind to that target protein. At this point, we don't care about function yet. Once we find those direct binders, then we take them through functional studies to see if those things not just bind to the target, but do they inhibit its function. And then we confirm that in vivo experiments through cancer phenotype. So this will be the outline of how I'm going to show you our efforts on CD99. We're trying to find a small molecule for CD99, just like Dr. Kim mentioned earlier. This thing is expressed in pretty much every single Ewing carcinoma cell. And earlier work by others showed that if you use an antibody to bind to CD99 from its extracellular domain, Ewing sarcoma cells did not really survive that. So there was promise that if you can inhibit CD99, it could be a potential therapeutic for Ewing sarcoma. It is a simple transmembrane protein. Unfortunately, cannot really draw anything below this because we don't know exactly how it works. So far, what we know is it's expressed a lot on Ewing sarcoma, and if you can block it, it seems to kill Ewing sarcoma cells. So what we did was we made this protein in bacteria, we purified it. Now we have a recombinant protein. We put a biotin tag on it so that we can capture on microchips for technical purposes. And then we confirmed that it was a good protein by using high affinity antibodies. So we can put this on a BioCore chip through the biotin tag and then run these low nanomolar affinity antibodies, and they recognized, so the protein was good to go. What we did was we used the same BioCore technology to screen small molecule libraries. So BioCore is a biophysical method. There's a microchip. You immobilize your target protein, and there's a microfluidic system flying small molecules over that. So here's an example of data coming from a 96-well plate. Each line represents a different compound. So most of them are not binding, but you have a couple of binders here. And when you combine the entire library, you get a picture like this. Each spot here represents a specific compound, how strongly it is binding to your target protein, and it is normalized for a binding to a non-specific protein. So at the bottom, you have your baseline, bunch of non-specific interactions, but at the end, we ended up about 100, 150 compounds that show promise that they may be binding directly to CD99. So once we have those 150 compounds that we think they're binding to CD99, our next challenge was to show how many of those are actually inhibiting CD99 function. For that, we took a comparative approach. These cell lines called U2OS are osteosarcoma cell lines. These are RDS and TCN1 are Ewing sarcoma cell lines. Osteosarcoma does not express CD99 at all. Even if you force them to express, it has the opposite effect. It makes them grow slower. So what we did was we compared the effect of those 150 drugs in these pairs how drug affects the viability of the Ewing cell line versus how it affects the viability of an osteosarcoma cell line. We picked these compounds in this area, so these are the compounds that represent the ones they don't kill the osteosarcoma, so osteosarcoma viability is close to 100%, but they kill the Ewing cells more than 70%. So this lower corner is our target zone. Those are the compounds we like because they're selectively killing the Ewings over the osteosarcomas. So that narrowed it down to these two compounds. We came out with clofarabine and cladribine. And the good thing I like about this one was there were multiple students working in the lab doing all these studies. One of them came say, oh, I found a compound that's here. It gave me the NSC number. The other student came and gave the other NSC number. We didn't know, but when we look at the structures, these were very similar. There was only this fluoride atom different. And having these two compounds coming from two different students was a really good confirmation that it was not just a fluke. And we went back to BioCore to confirm that with multiple doses, they do actually bind to it, and the affinity was around 3 micromolar. All right, now we have binders. What do? We went back and did hundreds of IC50 that you wouldn't really read from those. Here's a good summary of that table. All those students over the years, they made this incredible table. So here what we're looking at is an effect of clofarabine versus cladribine over 14 different Ewing sarcoma cell lines, 20-some non-Ewing sarcoma cell lines, and the bars are representing the IC50 values. So all these Ewing sarcoma cell lines, the average, they had about 0.5 micromolar IC50. All those non-Ewings, they have about 14 micromolar IC50. You have a similar thing with the cladribine too. So just going to a large number of cell lines, we were seeing the same specificity. These compounds were really more specific for Ewing sarcoma. Cladribine and clofarabine are FDA-approved compounds. They're actually approved for use in leukemia. So these were our positive controls. These are leukemia cell lines. Those were sensitive as expected. Then we looked at CD99 levels on those 40-some cell lines, and the protein levels of CD99 was correlating with the observed IC50. That was another good indication. It was specific. We also knocked down CD99 expression in Ewing cells, and when you get rid of CD99, the IC50 curves were shifting to the right. That also tells you the effect we are seeing is at least partially due to inhibiting CD99 on the surface. And we wanted to know, does this compound really do anything to CD99 on the cell itself? Because all direct interaction studies we have done so far was with the recombinant protein. So we took Ewing sarcoma cell lines and did a cross-linking experiment. So this is CD99 monomer. If you add a cross-linker, it goes up there, you double the molecular weight. And we added the compounds and look at how much dimerized CD99 was on the cell surface. So by adding drug, we were inhibiting dimerization of the CD99 on Ewing cell surface. So the drug was working on the target at the target cell. The drug was also inhibiting protein-protein interactions of CD99. This is a co-immunoprecipitation. CD99 comes down with a protein called cyclophilin A, but when you treat with the drug, they go down. So the drug was hitting the target on surface. However, just like I mentioned, these were FDA-approved compounds. Their mechanism for leukemia is inhibition of cell DNA replication. These are purine analogs. So they do block DNA synthesis. So therefore, when we give them to Ewing sarcoma, it is very likely that we could be killing those cells, just like those are killing the leukemia cells through inhibition of DNA synthesis. But we were trying to prove that it also may function through CD99 on cell surface. To prove that point, we worked with a derivative of clofarabine. This is a triphosphate version of clofarabine. Clofarabine triphosphate is not cell permeable, so it cannot penetrate the plasma membrane. So presumably, it won't be entering the cell and blocking the DNA synthesis, but if it is still binding to CD99, we should see the same phenotype. When we did the BioCore experiments, triphosphate version was still binding it with a similar affinity to the target protein. When we did the cytotoxicity experiments, in 2 different Ewing cells, the drug was showing the same phenotype as the regular clofuribin. So even the phosphorylated version that cannot enter the cell was still giving you the same cell killing, so it didn't really need it to inhibit DNA synthesis, but it was acting with the receptor on the cell surface. Again, to make the point, the drug is specific. One last experiment we did was we went back to osteosarcomas. Osteosarcomas do not normally express CD99, 2 lines. We can force them to express CD99. With that forced expression, they show the opposite effect. In osteosarcoma, when you express CD99, their growth is going down. 2 cell lines, the growth is going down. Now, by adding the CD99 inhibitors, these cells are growing faster. So we are reversing that inhibitory effect of CD99 in osteosarcoma. This, you wouldn't get a non-specific off-target effect of a compound that would make another cancer cell line grow faster. If it's a non-specific toxic effect, you would usually see cell death as an outcome of that. So making cells grow faster, you're reversing that negative effect of CD99 in osteosarcoma. And finally, we wanted to confer all of those in an in vivo study. Went to orthotopic xenografts. We implant tumors in Ewing sarcoma tumors, let them grow to 150, 200 millimeter cubic size. Then we randomize them and do a little trial in mice. We did that with Ewing sarcoma first by giving the compound IP. Clofuribin was working much better than clozapine. Then we repeated this thing by giving the same compound oral. We got much better results. So, and animals were treated only 2 weeks. So once we stopped the treatment, eventually tumor grows back. So we would need to go back and do a couple of longer-term multiple treatments. But this was a very significant improvement in survival and tumor size. And we repeated that with actually 3 different xenograft models. In all of them, clofuribin was giving better survival. It wasn't as good as the TC71, but we got basically the same data with 3 different models. So with that, I would like to stop here. Thank everybody who worked in the lab over the years, former members as well. And this was supported by CCF, and CCF also gave me great support in earlier years with our other work on osteosarcoma projects. I'm forever grateful for that. And of course, Jeff and Katja Scotlander from Italy, they contributed a lot to this project. And I'll be happy to take questions.
We actually have some time for questions.
So, this therapy that you're using, compared to what's used in AML patients, because the toxicity of the drug in those patients is really remarkable.
Right. So we were copying the dose that was used in leukemia models. So the dose was similar. And when I was talking to clinicians, so with this data, we are trying to convince people that perhaps this is worth doing a clinical trial with clofuribin in Ewing sarcoma patients, and that was the first concern. Everybody's concerned about their experience with clofuribin in leukemia, and they're hesitant to use this in a new indication. So that's why we're looking into some synergy experiments. So we're trying to do synergy, and we're finding clofuribin inhibition of CD99 shows some synergy with some of the tyrosine kinase inhibitors. So when we finish that, that will be one way of lowering the dose a little. And we have an NIH grant. It's going to go back this year. With that one, we are trying to make a more stable clofuribin triphosphate derivative. So if we can keep the triphosphate is not stable, it will be cleared eventually. If we can make one that stays only outside the cell, then you would not see most of the side effects of clofuribin that you're seeing because of the inhibition of the DNA. Brian?
Is there any like endocytosis type process happening? You know, like you bind on the. Sorry, is there any endocytosis type process occurring? You know, like you can give antibody, you know, conjugated drugs that way, and how they, you know, it's that they're endocytosed into the cell, and that's how they work. And do you know? So CD99 is broadly expressed on hematopoietic cells. is CD99 on AML cells that might explain some of its specificity to those leukemia cells?
Right. So the original mechanism for this compound, clofarabine itself is not the active compound. It's a prodrug. It gets converted to triphosphate version inside the cell through a kinase, and the triphosphate version is the active form. Leukemia cells are sensitive to this because they express high levels of the kinase. So that's how it went into the leukemia. But just this year, there was a paper, they showed CD99 expression in cancer stem cells of AML patients. So with that, perhaps the benefits of clofarabine in leukemia could be hitting both of them. It could be inhibiting DNA synthesis and inhibiting CD99 in them as well. As far as the endocytosis, I haven't seen any papers. We haven't done any work with that. So we don't know if CD99 gets through that endocytosis or if the antibody engagement accelerates or slows down that process. We don't have any data. Right, thank you.
Any other questions?
Oh, I'm sorry, there's one on.
Thanks. That was great. Just had a question. When you say inhibition of CD99, what activity do you think you're inhibiting? Is there the binding of an extracellular ligand, or is there some intracellular correlate of blocking CD99?
So all of that would be a speculation at that time. We are assuming that we are inhibiting an extracellular ligand, which we don't know yet. So we do not have an extracellular ligand we can put into the system, activate this receptor. We don't know what it binds to. There are papers suggesting that dimerization of CD99 itself, or maybe one CD99 on one cell to another on the other cell, activates each other. So it could be a ligand for itself, but we do not have a ligand, but we are doing experiments to find ligands for this. So that's one mechanism. The other set of experiments we've done a kinase array profile. So we know which kinases are up or down when we add the antibody. And we know which kinases are up or down when we add the compound. And we're looking for the overlap. So there are 3 or 4 kinases that are regulated the same way when we use the antibody to block the CD99 and when we use the compound to block CD99. So we think the CD99 functions through those kinases, but we do not know how CD99 activates those kinases yet. There could be 1 step or 3 steps in between because the receptor itself does not have a kinase activity.